Without wasting time, this month's guest is Dr. Florian Kuchenbauer. So Florian, he's my own supervisor, completed his MD at Ludwig Maximilians University in Germany, which is a very good school, and completed his thesis at the Max Planck Institute for Psychiatry. He obtained his Ph.D. At the Terry Fox Labs at BC Cancer for experimental medicine under Dr. Keith Humphrey's supervision, and started a lab at and now, up to this present day, with his wife, Dr. Arefeh Rouhi, opened up the Kuba lab on the 13th floor of BC Cancer Research Center, the Terry Fox lab, and the lab which I'm in today. So, welcome, Florian. How are you today?
Thank you. So to remind you, this is the lab, which is the noisy lab on this floor, right?
We are the noisy lab. We are the very noisy lab. The most social lab, though, most fun lab.
That's a nice way of putting it.
Yeah. And so, you like when I first started this idea for this interview, you were actually one of the first guests I had in mind, because out of all the supervisors and all the scientists I see in the building, you are definitely by far the most passionate and enthusiastic person I see all day. Every day I see you, you're high energy, you have quick strides, and you zoom buzz past the clinic and the lab, and no matter how busy you are, you always sort of have time for us, and you're always bright with a smile and joke around. And so this is more so curiosity from my part, but what made you so passionate about this work that you do, working both as a clinician and as a scientist? Because you mentioned early on that you wanted to actually become a chemist, right?
Yes, it's true. And the reason I think is ADHD. So that's something which is extremely helpful. And there was actually a really good article in the New York Times or Economist, I forgot, about how ADHD is helpful in a pandemic and uplifts people to forget about their anxieties, about all the misery that happens around us. So that's one part. The second part is that I've been always very curious by nature. So when I was 10, actually, I told you about it, but the biggest wish I had when my dad asked me, like, what do you want to get for your birthday? And it was not a Super Nintendo I asked for, because it didn't exist at that time. But I said a chemistry set. This was like the thing I really wanted. And my dad was like, this is an odd kid. But he bought me a small one. This was like the first time that I blew up something in my bedroom, and my mom was not happy about this. So so then I asked for a bigger chemistry set afterwards, which took a little while to persuade my parents that this is the right thing. But this kind of interest, and this was also the time where we actually read books rather than being on Instagram and Twitter and stuff. And so I spent a lot of time in libraries. I was pretty nerdy at this time, and read a lot of scientific books. And it was always kind of an interest. So I don't know why, it's intrinsic. But as I said, like, I never considered about going to medical school, and then I got into chemistry after school. So I studied chemistry, and I hated it right away. It was horrible.
Yeah.
It was really horrible. I don't know. So I had to think about something else. My mom told me, like, why don't you think about medicine and medical school? And it's very difficult to get into. It's everywhere the same problem. And myself, I wasn't really convinced. So there were two factors that really kind of made this decision or drove this decision. This was one factor, was that I read a book. It's I don't know the English title. It's called the Medicus. Like, it's by Noah Gordon, and it's about a guy who lives in the medieval ages, and then he travels to the east to become a physician. And I was like 19 when I read this book. I was highly inspired, and I was like, this is what I want to do. This is cool. But then my parents said, like, hey, maybe it's not exactly like this medicine. So maybe you should work in a hospital. And so I did I asked, there was a hospital close by, and I asked the nurses if I could come and join them to work for free for like 3 months, just to get an impression. And they are like, oh, okay. So this was like the first time in my life when I had to get up at like 4:30 to start at 5 o'clock working. Kind of stuck with me, but at the beginning it was really hard. And you know, and the first thing, like my first day, the nurses were like, "Hey, Florian, look at this guy in room 200. Like, is he well? Can you check on him?" And they sent me in there, but the guy was dead already, right? So they made a joke with me, and these things you cannot do anymore. I know this, but this was like
Yeah, yeah,
1992, and so this was like my first how's like birth by fire, is this what you say, right? This is
yeah,
like
yeah,
how
yeah.
what they do with you at the hospital in these days, at least. And you know, it was actually, it really grew on me, and I really enjoyed working there. And that's how this started. And then I applied for medical schools and was lucky to get into Munich.
Interesting. So, I mean, is it your sort of interest in hematology that came along during medical school or
It was
during
actually
residency?
cancer. Yeah.
Cancer as a whole.
Yeah, hematology was not this kind of came later on. I so I mean, what do you do when you go to medical school? You have to study, and you also look for like parties, and you realize you can also go abroad with medical school, like with science. And this is something I was really interested in when I applied at the Max Planck Institute, which was also like a very renowned, or is a very renowned institution in Germany to
Of
do
course.
research. And I applied to do a medical thesis, which was an experimental one on pituitary tumors. One of the first things I asked them is like, is there a way to go abroad? And they said yes. And my supervisor was from Argentina, the Ph.D. I worked with. And he set me up to get a scholarship to go to Buenos Aires for a few months to work in a lab there. And I was able to go to Italy to work in a lab. This was all like before 9/11. This was all in 1999. So you can imagine when I went there, they gave me like cells to transport. And the way they did is like, hey, you have to keep cells warm. So we had these Falcon flasks, like the small ones. And what they did is they were filled up with DMEM and a red fluid, and then they attached them to my body for this long flight to keep them warm. So if you do this nowadays at an airport, this wouldn't go well. So this is like how I traveled to Italy
Like
to
bioterrorism almost.
Argentina, and with like vials attached to my body, which is like I didn't even think about it. I had just had a letter from my professor saying like, this is fine, Florian is a medical student, blah blah blah, and he's bringing cells over. And if I think about this now, this would be maybe not so good to do this.
Did you manage to keep the cell alive here?
Absolutely. I brought them over. I even remember the name. The cells were called ATT20 cells, like pituitary cells. I remember this because it was such a, was very unforgettable.
So you did an MD thesis. So was that sort of a mandatory thing for your medical school? And was that sort of what kind of propelled you to do research in the end?
Well, in Germany, to get to call, so there are two things, right? In Germany, to get an MD degree, like an MD, you have to do a medical thesis. So you can call yourself only doctor because it's becoming a part of your title. It's a little bit like a German, Austrian, Swiss thing. You have to do a thesis, and you can do it. You have to do it while being in medical school. And you can choose whatever you want to do. Some people do like something statistic and small, and some other people do, I want to do something experimental. So I wanted to learn how to work in a lab, and that's why I picked this place to be. And yeah, so that's why I'm, yeah. In here, I think when you finish medical school, you're an MD automatically. You don't have to do this kind of.
No, there's no thesis
thing.
part, right? So,
No.
And so, is that like your like early experience working in a lab sort of what kind of pushed you to do a PhD as well on top of an MD?
No. And I actually didn't want to do a PhD initially. So this was when I came here, and Keith, like I had a postdoc scholarship from the German government to come here to do research here. And so Keith said, why don't you do a PhD? And then I said like, no, I mean, because it takes long, right?
Yeah, exactly. Yeah.
And I asked people in Connie's lab, and they're like, oh, I've been here for 6 years, 7 years. I was like, maybe not a good idea. Like, especially when I still was in the middle of my training, right? So I had finished 4 years of internship and rotations, and I still had to do another 2-3 years to get my board certification. So I decided on not doing this, but I was really fortunate because at that time the director of the experimental medicine program, his name was Vincent Duronia, like super nice, super supportive. He kind of gave me like a pass to skip all my coursework and just do the comprehensive exam and the final exam, so I could actually just do research. So this is the reason why I could pull it off in 3.5 years.
Oh wow. And,
Yeah.
you know, you always like tell us during your time doing your PhD about how you sort of came across microRNA research and how you initially wanted to study microRNAs, but Keith was like, ooh, like sort of reluctant, and how you sort of were able to detect them and show that they were real. So could you go over again how you were able to, like, during your PhD, were you able to detect microRNAs? Because I thought that was a really interesting story.
So before I came to Vancouver, I applied at multiple labs. So I applied with Dominique Bonnet in London. I applied with Thomas Look in Boston, and with Keith. So I interviewed at all three places in one go. It was like one big trip, and the first thing was that I talked. So the reason why I got into like non-coding RNAs and was because there was a paper in Blood in 2004 about the gene called PV, and PV is and nobody knew what it was. So this is something I started working in a lab in Munich with Christian Buske, and to be honest, I was a full-time clinician. It was difficult to do any functional work. It was very limited. So I was working with Thorsten Haferlach in Munich about like FLT3 detection in patient samples. So this was much easier for me to actually produce papers. But this PV gave me like, I started reading about this gene, and people suggested that this is relevant for non-coding RNAs. And to be honest, I had no clue what this was. And then I started reading about this topic, and then at this time, the name microRNA was not very common. They were called like sTRNAs, like small temporal RNAs. They had different names, and I proposed kind of a project of looking into this at different labs, and they're all like, we don't know what you're talking about. So, but that's kind of Connie, and this was one of the key ones. When I interviewed here, I also interviewed with, like, not interviewed, but Connie took time and spoke to me, and which was something I always remember, because it was really good, because she also told me about like the difficulties she's working with, about high publications. What does it mean, high impact, low impact publication, is how relevant it is. So anyway, so I was, so this was a positive experience. And so Keith eventually took me, which I was very fortunate about. But he was on a sabbatical during this time when I started. And so he wasn't there. And I had like, I started thinking about this project. So I tried working on this PV gene. I couldn't get it cloned. And then I moved to a gene called like to Argonauts, like AGO2. I tried to clone. So didn't really work, these things. And then microRNAs became like, there were first papers popping up about microRNAs at that time in AML. And I said, like, I have to, and this is what I want to do. This is interesting. So Keith said, like, we sat down when he was here doing a sabbatical for a couple of weeks. And he said, like, prove to me that these microRNAs are actually existing in leukemias, because nobody knew. It was completely unknown. And I was like, why don't we do real-time PCR? Why don't we do an array? And there was no sequencing at this time. Like, this was the pre-sequencing, like next generation sequencing time in 2005, 6. So, and I said, like, okay, so how should I do this? And I mean, I'm a physician. I had no clue how to do this thing. So I had a lot of support in the lab. I have to say, like, I had people with whom I worked with, and these are friendships that I still have, which is really good. And I also had an extremely nice technician who was the heart of the lab. Her name was Patty Roston, and she was amazing. She helped me so much with these molecular things. So I came up with I found a protocol to clone microRNAs from plants, and this was a ligation. Basically, it was a race PCR. I don't know if this technique is still familiar with people, based protocol, and I adapted it for my purposes. And it took me like half a year, and then at the end we took like these cell lines Keith had in his lab, and I had to do, I think, 500 to 1,000 mini preps just to, and then suddenly we got like sequences, and they came back. I remember this was in August in 2006, and these sequences came back, and suddenly they matched to microRNAs. I was like, holy shit, this is like, I can't believe it. I was like, this can't be true. This was really like amazing. And then I showed to Keith, and he's like, I was like, I found microRNAs, and he's like, Florian, do you know what you just found? Like, do you know what this means? I was like, I have no idea. I mean, they're microRNAs, but I think it's really cool. Can we continue working on this? He's like, yes, like we should do this. And so this was basically a starting point. And Keith was fantastic at opening doors. Like, so he hooked me up with Marco Mara and with now what's his name, from Upsala, the head of Upsala. I'm just stupid. Anyway, with
Oh, I can't remember the name either. From offending anyone.
help me please. Yeah, but anyway, so first he hooked me up with Marco Mara because, and at this time, they got the first next generation sequencing machine, which was called Solexa sequencing machine at this time, right? And they were looking for projects, and they want to do a microarray project. Carl Hansen, yes, Carl. I'm silly.
Yeah, there we go.
Looks like me, but I can't remember. Silly, huh? So Marco had Ryan Morin at this time working on this, who was a PhD student, and Ryan and we sat together, and I mean I was pretty useless, to be honest. I did all this work about micrornas, but he did all the sequencing stuff, like all the bioinformatics behind it. And so and then Connie also had a good project. So these were two projects, one in ES cells and one in leukemic cells, which were done back to back, and they were both published the same year in Genome Research. And this was basically these two were the two papers where isomers were discovered. So these are isoforms of micrornas, and Ryan and I were sitting in a cafe across the street, which is now I think it's called Korean Cafe now or something.
The Korean Cafe, the K Cafe,
yeah. So we're sitting there, and he's like, "Hey Florian, I found this thing and I came up with a cool name. What do you think about isomers?" And then we're discussing, joking about it, and we said like, "Hey, we should use this for the paper." And this is kind of how this name came, and you can find it on Wikipedia now, which is kind of funny, but yeah. And so this is how this name was invented. And interestingly, when you read the Wikipedia article, there's no mentioning of like Ryan or me or so, like bringing up this name or in big time. So it's kind of funny how these things sometimes get attributed to other people, but it was actually us and especially Ryan to come up with this and to publish this the first time.
Interesting. So, I mean, I guess that's how you sort of got your big break in starting like your own niche and starting a lab in the future. And so, after your PhD, how did you sort of end up in setting up a lab and working at Ulm?
Well, I should say like so the guy who was so Christian Buske, with whom I worked in Munich, he got a big position in Ulm, and he introduced me to the head of the hematology department. His name is Hartmut Döner. So this guy is one of the biggest guy in the world on AML. And so, for example, he's the one who he worked first on CLL, then on AML. Now he's pretty much well known. And I met him at ASH then and got introduced. And he said like, well, you can start in my clinic. And that's where I went to Ulm. And when I went to Ulm, and I took really nice pictures of the houses there, like these old houses. I wanted to make it like to sell it to Arefeh. So like, this is a beautiful place to move to, and coming from Kuba, this is a hard place to beat.
It's
if you go somewhere else.
To leave, right? Yeah.
And so we eventually moved to Ulm, and then let's put it like this: it was not as so we didn't get a startup package. So they said like, we can give you a room. The room was the garbage room of the other labs, which we had to clean up and we had to get renovated. I'm very honest with you. I mean, we didn't get any money from the faculty. I just got half a year off before I went back to the clinic to write papers and proposals. And Arefeh and I wrote a bunch of proposals, and we were lucky to get funded. We drew in a lot of money at the beginning so we could start our research work, but I was never, so Arefeh did her postdoc with somebody else, with Christian, and then with another group at that time. Mm-hmm. So she was not 100% dedicated. I wasn't. I was then seeing patients 100% of my time. So everything we did later on was we had to bring in money ourselves. So I had a lot of like sleepless nights because I was worried we couldn't get the money and couldn't get science starts, because otherwise without money you're done, right? That's the problem.
Exactly. Yeah.
And also like the time, because I never had dedicated research time. I mean, here in Vancouver is actually the first time that I have like a split position, which is great. And in Germany, it was we had lab meetings in the evenings at 8 o'clock at our house with our students, because during the day I was just doing rounds in patients. This was not so easy. Like, not everything was easy there, but we somehow managed, and now we're here and we are fortunate.
So, I mean, going through all that trouble and sort of struggling with funding early in the beginning and having such a rough start, I mean, how did you sort of manage to sort of find your ground on continuing research? I mean, if you're working at the clinic constantly, doesn't the thought pop up where you're like, what's the point? Why would I struggle opening up a lab?
I mean, honestly, first thing is Arefeh, right? She
Yeah,
managed this thing. It was her. She put in a lot of her time besides doing 2 postdocs to run this lab, and you see it's the same. It's still the same, right? So without her, it would not be possible. I mean, this is kind of the beauty of our of just working together. This is really it's for me. I wouldn't be here without her. It's very true.
of
and
course.
and so this is a good thing. The other thing, of course, is like so this is the physician perspective on things, and this is something not only me but every physician in this program or in any hematology program, especially with malignant hematology, has to deal with, right? You have to sit sometimes in front of people who are maybe 20, 25 years old, and you have to tell them, I'm sorry, you relapsed, I don't have any other drug, please get your papers into order, please. And then they have 2 small children, and then once I had to sit in front of 2 10-year-old children and tell them that please stay in the hospital, your dad's going to die within the next few days, you can stay here, but these are the things this is something that drives me incredible. Like, this has really shaped the way I think about science and how science should serve the community, and how I want to do the science we do in our labs. So that's why I have this this intrinsic drive to really make this a better place. I don't know if it sounds a little bit cheesy or so, but it really is. I mean, I had this conversation yesterday with a young guy. I have this conversation tomorrow with somebody else who is like 35, has a daughter who's 7 years old, and it's very, like, there's no justice in disease, and there's so you have to kind of find ways of improving things for people. I mean, there's a reason why AML has a overall survival rate of, I don't know, 22% in Canada.
Yeah, it's quite low. And, I feel like for us as well, you having your foot in the door in both sort of careers in science and, in the clinic, it gives us students so much more insight too. When I can read as many clinical papers as I want and I see some survival curves, I'm like, okay, well, this drug does this drug does that. But when you come to our lab and you talk about, this patient took this drug, which we know about, and, did poorly or relapsed, or, this patient had remission and it actually worked. It gives us so much more weight as to what we're dealing with, right? And sort of the drugs that we're testing and what we're looking at, and sort of, the cytogenetics of AML that we study, it has so much more weight in terms of that, because you come and tell us this, firsthand experience. And, when we would look at a paper and you see, oh, this drug has, much better use than the other one. And, let's say 40% survival versus 20%. And you're like, oh, this drug is great. But then you come to tell us you're using this drug, let's say midostaurin, and this patient relapsed and is not doing well, likely won't survive. And that gives us so much more insight, being like, okay, this drug needs far more improvement, or like this really needs some improvement. So, yeah, we, and
but you see, this is exactly what you mean. So science has to have, in my eyes at least, has to have an emotional aspect to it.
exactly.
And this is the driver, at least for me. This is the way I'm structured as a person. I have this emotional core, and every time I have to deal with this, it tells me, you have to work harder. You have to do something about this. I mean, I got hired, or like Aref and I got hired through philanthropy to come here to bridge this thing, to establish a bridge between clinic and research. And we have here fantastic clinicians. Like, this is a great team I'm working here. We have there are always this problem of limited resources. We cannot afford this drug or that drug. This is in every system. And then you have on the other side, like just a block away, you have researchers who are outstanding. Like the TFL is and in general the Cancer Research Center is a world top class research institute. But the communication between, like the block in between, is like, sometimes it's hard to, it's like an ocean sometimes, right? Because people don't talk to each other, and they're
Mhm.
and I really hope we can and there are other people like Kevin Hayes doing exactly the same, and who we really want to bring these two things together and try to what comes out of it to the community, to patients here in BC.
Exactly. And, you sort of, and, you having this sort of marriage, not just in life, but in science as well with RFA kind of gives us insight to both sort of, clinical perspective and the molecular perspective. So we have sort of two different reviewers sort of looking at our data constantly. And I feel like that really pushes us as well. And
but you also see that we have different aspects of looking at things, right?
exactly,
So RFA
yeah.
is really has the research biologist aspect, which is extremely helpful, and especially, for example, now these NRIP revisions. I mean, honestly, gene regulation. Who am I? I'm a physician. I just don't know these things. I have to learn. I didn't know what high C is. I have to read about this. I have to learn about this, right? This is not a given. I have to be up to date on clinical trials. I have to be up to date on research. I have to focus. I have to know something about every project we're working on. So it's really great. But I have this aspect where I can say, like, you know what? They just don't do well, these patients. I mean, we do something, I see these people, and I also see positive aspects. I see like drugs. I have patients who relapse and they are on gilteritinib as a drug and 3 pills a day, and they are treated, they're in complete remission. I mean, amazing. So
Yeah.
you have both sides, and this is the beauty. So you get, as a PhD student, you get, or anybody who works with us, you get all these aspects from us, and I think it's a good mix.
Yeah, I mean, we're like unintentionally advertising our lab here, but this is something you always talk about as well. But what is sort of the potential that you see in your lab, and where do you hope to go with this lab in terms of both, in terms of clinical benefit and in terms of pushing what's happening at the lab towards your patients?
This is where the students pay attention, please.
Jumon, see, the things like this. There are several ways of. I've been thinking about this because, like, I reflect. Like, when I came here, I had a lot of discussions with, like, Keith Humphreys, who's my mentor, and about what is relevant. And we had these discussions before, also with other people who run labs. But what I think, what I learned for myself, the most important thing are the people. And the reason is, so for example, this paper we published on micrornas in 2008, I mean, honestly, I don't even know if the data is right, because the technology has developed so much. It's maybe a milestone for something, but it's not something you would use the data for anymore, because the turnaround for new data with new technologies is like maybe 5 years or so. So this is, nobody cares about this. There are very few papers who actually have long-term implications, considering all the papers that are published currently day by day. But so what I think is, I'm quite happy at where I am, and I mean, my goal is, of course, like the things we do in the lab: find drugs, bring them to patients. I want to build a phase 1 unit here where we can treat new drugs, and then these things could feed into our science to figure out, okay, what are resistance mechanisms, how we can evolve these things. And I would like to do personalized medicine by testing drugs on patients individually and then trying to treat them. These are things I want to do. But at the end, what counts is when you guys come out of our lab and have some of the thoughts we told you about, and carry this on in your life of research, and maybe go somewhere else and tell it somewhere else, and have the spirit. I mean, it sounds also pretty cheesy, but I really believe that this is how science actually, this is what is important. And this is how science, this is what drives science. Like I think a lot about the things Keith Humphreys told me. Like, he asked me once, like, I asked him, like, we had a discussion, this was in 2007, or 8, and a paper got rejected from Cancer Cell, and I was so frustrated about it. I was so angry, and I was in his office, and I told him that this is not fair, and I think this is the bullshit reviewers and blah, like, all these things we always say about these things if something doesn't work out. But then he looked at me and said, like, hey, Florian, do you think I'm a failure? And I was like, no. But he's like, I don't publish all the time in Cancer Cell or in Nature, or I publish in experimental hematology, I publish in leukemia, I publish in blood, and like, but do you think I'm a failure? And I was like, no. And
Mhm.
so it made me think about this, what is relevant. But and this is something Connie also told me once at my interview in 2004. She said, like, even if you don't get a high profile paper, the data we produce is solid, the data we produce is reproducible. This is like good science. And these are the things, right, I kept for myself. And he also told me, like, because I asked him about advice, what is when I before I left his lab and asked him, like, what is kind of a supervisor, what should I do? Can you give me advice? And he told me one thing, and I always repeat this because I think this was the most important advice I really got, is like, he told me that your job as a supervisor is to bring the best out of people. This is your job as supervisor. And it's so true, right?
Yes.
And I mean, and everybody, so when he retired to make space so Aref and I can start, everybody said he was a great guy, great scientist, gentleman, only positive. Nobody said, like, oh, he hasn't published in Nature, oh, he hasn't published in Cancer Cell 50 times a year, right? This is not the perception. The perception is what you are as a person and how you, and I think and that's why I'm quite happy about this, right? So I think we're doing well in our lab. We have a extremely nice lab. We have people have the spirit. They want to do something. It's good. I mean, that's all we can do.
Yeah, and that's the advice I won't forget from Manny as well, my previous supervisor, where I was looking for supervisors. I'm like, Manny, what should I look for in a supervisor? Like, who do I choose? And Manny was always like, it's not always about supervisors picking you when you submit your CV. It's about you picking your supervisors. And he said, pick someone who's hungry, pick someone who brings the best out of you. And I mean, that's why you always tell me, you're like, oh, why did you pick me? Why did you pick me? I'm like, well,
no, I don't always tell you this, and I don't always ask this.
Well,
this is not true. I asked you once.
You told me, you mentioned it before though. You're like, and you asked me before, you're like, oh, I always wonder, like, not always, but like, I wonder why you picked us, like our lab. And that's exactly why. You sort of have this sort of hunger and you sort of passion for research and this sort of wanting of, me sort of bring the best of myself, sort of me doing my own thing. And you just sort of, you both being sort of there to push me. And I think everyone in our lab can speak that the biggest thing that we appreciate is how much you sort of support every member of us in the lab in terms of both our academic life and our personal life.
Because I tell you, don't take breaks, don't skip lunch, and stay late.
Yeah.
This is like supporting your private life.
Yeah, those subtle.
All the Kuchenbauer kids late at night, having dinner together, but at the same time looking forward to other 5 hours of work.
So yesterday, when we received your Western blot at 1:30, I think, or so, I can't remember. So this is like, I love that. I was like, this is the spirit. Like, they're in the zone now. This is good.
But like, you need to believe us when we tell you it's not because you sort of leave subtle hints of telling us to stay late. We all know that's a joke. And if we took that as a joke minus every other aspect, we wouldn't be there. So the biggest thing that sort of makes us stay and the biggest thing that sort of pushes us to be there is you both, Florian and Arefeh, always supporting us and always being there. I mean, if we need like a 10 p.m. or 11 p.m. meeting, you're there. Last minute edits, you call me at like 11:30 p.m. I'm like,
yeah, first of all, I complain to you about last minute edits. That's the first thing I do.
That's true. But
this is the thing, right? So this is a fun part, right? I don't have a contract that says I have to produce this many papers, get this many grants. I could also get a season pass in Whistler and just enjoy that. To be honest, there's enough you can do here in this beautiful environment, but yeah, it's also like, for me now, I have also the advantage of working with a great clinical team. Like, for example, I work here with the hematology research program, and in particular with Nazim. I think she's also on the call here. And she runs the first clinical trial where I'm PI. Like, it's an industry sponsored trial. She basically does all the work, right? So but it's really, so this is the fun part, right? You do all these different things. I can see patients here and think about clinical trials. I can work with you guys and think about how we can, the things I see here, how we can make them maybe happen in the lab. And then you have Arefeh, who is more focused on the lab side, but we discuss a lot at home about, you know, clinical aspects of our work, and this is the fun part. This is the best part of it, really. It's this mix of things, and I, you know, I'm still not tired of it. I mean, we just started here 2 years ago.
Yeah, exactly. And you're not wrong about, you know, leaving sort of an impression first before anything else, instead of trying to publish. I mean, if you ask any member of our lab, the first thing we say is how much we can rely and we can trust you both. And I think that's more important than.
So what you want to say with all these things is, don't work at Stem Cell, come to our lab. Is this what you want to say here? that sounds a little bit like it.
Yeah, you're going to be flooded with applications soon. Your email is going to be.
No, but you know what? And this is the great thing here in Vancouver. You have a great community of a lot of different people. Like we got approached here by different people to work with them. Like we were basically welcomed with open arms. And before I started here, they made a schedule for me, and I asked them if they could, like this was in February 2018, if I could meet with Carl Hansen, with Carl again, because I haven't seen him for a while. And we had a couple of joint papers where we worked quite close together, and he always has good ideas, and we think along the same lines. So we met at UBC, had lunch together, and he's like, hey Florian, like if I would start a lab now again, I would reboot my science completely and think about things. And I was like, that's exactly what I want to do, right? We bring some money, do different things, not only micrornas. It's really that's what we do now. I mean, you work at most of these projects are a little bit out of my comfort zone, which I quite enjoy because I learn about it. And,
yeah, exactly.
And this is the fun part, right? And of course, we were very fortunate to be funded. I mean, this is, as I said, like I can't stretch this enough to how generous people have been with us when we came here, despite this really tight situation in Canada about funding. So that's why I, you know, share data. I really want to work with people. I think this is important.
Exactly. And, you know, I feel like these constant collaborations, I mean, including with my own project, so many different sort of, it's like little teams instead of individual sort of work. And so, you know, I feel like that really sort of helps out, and each student sort of gets different perspectives from different groups instead of just sort of a monotone, you know, monotone discussion with the same people over and over and over again. So, and I'd really like that too, you know, collaborating with SFU, collaborating with the proteomics and all these different types of things. So
yeah, this is good. Yeah, but you know, the thing I can tell you is I think you really have to enjoy what you do. Even, I mean, there's always pain. It's always blood, sweat, and tears every time, exactly. Regardless. And this is part of it, right? You know, pain, no gain. I mean, this is what it is, right? You grow with your with the things you do. And exactly. and it's the same for us, right? I don't think we will ever produce 50 Nature papers or anything, but it's really it's important about, you know, we enjoy what you do, we're passionate about it, we try to relate it to actual real life problems of real people, not to relate it to like log rank curves which you see in journals. That's what it is. And so I give a lot of quite frequently I give like interviews, not interviews is the wrong word, but like science updates for the BC community. So this is something we do. Mani and I, we just signed up for one in May, which we're going to do together. And these are things where you have to involve people and bring them together. And this will really help, I think, to not only to get funding for like special things, because I mean, building a hospital unit or something, this costs maybe $10 million, but you know, you have to make people aware of what you're doing.
Exactly. Yeah. All right. Well, thank you for so much good insight. And I hope a lot of people sort of find a little bit of motivation, a little bit of drive. I see our lab is sort of flooding the chat here. So we're going to head into the questions section. So if anybody has any questions, please put it in the chat, or because this is not a webinar version, feel free to raise your hand and unmute yourself.
Yeah, unmute yourself. I'll decide, I guess, like who to unmute if a lot of people want to say something. I'm not seeing a lot of hands. I do have a question. Would you say, Florian, that you customize your teaching mode per student? Like you teach each student in a different way, like according to their
Well, I can tell you that
learning.
Jumum needs a firm hand. This I can tell you.
Yeah, of course.
No, well, I don't know. You know what? Honestly, it's the same when you ask like, how do you talk to patients, right? You sometimes you get a vibe which direction these things should go and what people expect from you and what's important to them, and then you adjust them. And it's same way they approach me, right? They not always talk the same way to me, but I think this has already started in the selection process. I like how this lab came together. So we also had people who were not happy in our lab who left the lab, and this is also it's a selection process to a certain degree. And this doesn't mean that these people were bad or something. It's just sometimes it doesn't fit. It just happens, right? This is normal. Unfortunately, like pretty much everybody we have worked with is very talented. I mean, and sometimes as a supervisor, you cannot just make them better because it doesn't match. Maybe you don't have the same like vibe or like the same connection as to other people. I guess that's what makes us human, right?
Yeah. I have another question actually, but how do you balance the emotional stress between managing all of your students, managing the research, and then talking to patients in clinic? That is an overload of emotions. Do you compartmentalize, or what are your tips to keep like the stress underwater?
So first of all, Arefeh takes a lot of the stress. So she just, because she does a lot of these things and deals with a lot of things which I don't. I mean, it's very clear that this is so this is really something I have to stretch out. This is a joint lab. Otherwise it would not be possible and it wouldn't exist in its ways. So honestly, I do sports. Sports is the outlet for me.
Great. Thank you, Arefeh, for supporting the Kuba lab too.
She is the Kuba lab.
Yeah, it's part of the name.
Okay, so I do have questions now. The first question I might address is, what would you say is the comparison between the research here and the research in Europe? Do you think there are any differences, or can you say which one is better, or just like which one is different, or like how do you think it differs from one from another?
So I can give you an example, like in Germany, in Ulm, where we used to work, the system is different. So here, what I enjoy here is that the hierarchies are relatively flat here. So Connie, I mean, the first time I saw her, I felt like a little mouse in front of a cat because it's Connie Eves, right? The name is like, and I was like, Dr. Eves, and she's like, I'm Connie. And this is kind of, in Germany, you address people by their last name. You always call them professor, doctor, whatever. You have this kind of steep hierarchy, and which makes sometimes communication a little bit less personal or sometimes difficult. I think this is easier here. I mean, if the guy who gives me my driver's license calls me by my first name and says, hey, this is awesome, you got this driver's license and you're from Europe, I'm like, great, good job. I think this was very encouraging when I came here to see that. And the other thing is the level is the same, right? You go, there are good and bad labs everywhere, and you find every kind of research level. But I feel that here, what is an issue here is the funding system is much more competitive. I mean, funding rates are 10% here if you submit a grant. In Germany, we had funding rates of like 30 to 50%. So Arefeh and I got pretty much every grant funded we submitted. I mean, sometimes they didn't fund it and then we could resubmit it. We don't have deadlines to submit grants. You can submit them anytime for most funding agencies. So they meet like 4 times a year, these committees, and then distribute them to reviewers, but you can submit them anytime. So you don't have to wait until like in February or in March where everything's getting submitted, which is really stressful here. I find this pretty stupid, to be honest. As a system, it's not productive. And this is something which I find is better. What you can't do in Germany, you cannot submit the same grant or similar grants to 2 different agencies at the same time. Here you can do this, which is because of the low funding rate. So this is better. and you can recycle grants better here and work on them. So that's good. So, you know, pros and cons. And the other thing is opening hours are here. This is great. Like you can go to a restaurant here. You have 24-hour restaurants. In Ulm, we were on a hill. There was like the only thing that was open after 8 o'clock was like this little machine where you can put in like €1 and then you get like a Snickers out of it, right? So this is all. And here you can get some sushi in the evening. This is go to Cactus Club right now, maybe not, but this is also good.
Flexible hours.
Yeah.
Okay, awesome. I'm going to go to the next question. So how well would you say does the public understand the research and the progress being made in cancer research? Do you think there's a barrier for like the public to access and understand the knowledge about their own cancers in the clinic? And how do you think we can improve that communication?
I think this is a good question, and I really believe that, and that's why I do it. It's not to go out and involve people like the public who pay money into or fund research, to give them like an update on what we do, to give them like a insight, because people just don't understand how research works. It's complicated. I mean, this is something where we have to have more interactions, I think, with the non-scientific community to communicate what we do. I think this is something I would promote more. I think this is really important to and then people can ask questions. I mean, this is and there are no stupid questions. People ask questions and you try to answer them and see. And you know, I think the other thing I think is really important, to give a positive vibe to people, that research is important, that what we do can make a change. It might not be tomorrow, might not be in a week, but it will eventually, you know, you discover something, it might, it's going to get ignored for 5 years, and then somebody picks up on it and suddenly develops a drug from that. I mean, these things you cannot predict, and this is important. So this is positive.
Sounds good. Okay, and last question, I think, in case other people want to talk, but how do you balance personal family life with so many responsibilities?
There's skip the dishes, Uber Eats. It's like, no, honestly, as I said, like Arefeh and I are a team, and this is the way it works. So the beauty here is I don't have, in Germany, for example, that's another thing. I was on the ward all the time, so I had to start at 8 o'clock. This is when we start in Germany to work. And you stayed at least till 5. So here I'm a little bit more flexible, so I can take my son to German school on a Tuesday in the afternoon and then work from home or come back to work. Or so this makes it a little bit easier for me to do these things.
Sounds good. I guess that's all for the questions. I might have one last one. Would you encourage your kid to study science and to get into research? I think it's an interesting question to make a lot of scientist parents if they want to make their kids study science or technology. I
think the first thing I would tell them is like marry rich, and then you can get into science. Then you can get into science. This is the way I would do it. But he is yeah, why not? There's nothing wrong about it, right? But you have to be have to have the right mindset for it, right?
Yeah. Okay. Any more questions? It's a one-time opportunity to ask Florian anything. Anything. Not even lab members.
I'll ask a question.
Okay. Oh, gotcha.
I was just waiting for everyone else to have their turn. No, I actually, I'm kind of going back to your point where kind of as a clinician, which I thought was, I hear this a lot from clinicians who kind of identify certain areas for research, you know, as part of their clinical practice, and kind of how, I guess what my question is, kind of from an institutional kind of perspective, how do we best support, encourage, help identify what early kind of researchers need to know, and how do we best support them in that sort of crucial step from a clinician to a clinician scientist? Because, you know, like you said, in your clinical practice, you see all sorts of things that could be improved, and processes, and you know what I mean? And so how can we best support that threshold in those early kind of research years, that they go from kind of doing it from the side of their desk to kind of really building that capacity where they can then build a lab and kind of grow their own research capacity?
Um, I think the most important thing is, first of all, to get good mentorship. So I think an early on scientist should be paired with somebody. I think UBC has such a system, but I'm not sure how it works. But you need, in the clinic, for example, here you have to be attached to somebody who is a little bit ahead of you, so you can, so they will tell you what mistakes you can avoid, like which career options you have. But also working with this person, they might tell you, you know, maybe you're more or less suited for running a research lab, these or doing whatever it is. So I think mentorship is I think is one of the most important things, which is still not fostered enough. The other thing, of course, is you have to. So the problem I always had is that I never found time to do research in a good way. So there has to be a program, a dedicated program for clinician scientists, where people are getting, where they do not get paged, where they are getting taken out of the research, out of the clinical program to do actual research, because you need time for it. Otherwise, it's always mediocre. You cannot focus on something. So it's basically mentorship. It's dedicated time. And the third thing is you have to, and if you identify eventually somebody, you have to give this person an opportunity to get grants, to support this person, and to have a space to start a lab. So these three things, I think it's about mentorship, time, and money. I mean, this is, I think, this is what you need to really build the next generation.
Yeah, that's a great answer.
I know.
Wow, Florian.
Don't forget your ego. That's got to be healthy too. But you're right. I mean, it's about protecting that time, right? And that's with money, and that's with grants that are specific to kind of that seed sort of funding of that research.
But you know yourself from yourself, right? You people want to do clinical trials, and it's so hard to do. It's so difficult to raise money for it, to get support for it, to have somebody who tells you how to do it. It's really challenging, and it's involved, and it's connected to so much pain to do these things that, you know, people give up at some point.
Absolutely. And I unfortunately do see that, especially in such highly regulated areas such as clinical trials. And it's, you know, when it's your first few studies, it's overwhelming, and you need someone to kind of help sort of that orientation or that education or training, or I don't know, we could be better at it. So I hope we build that capacity as well. Thank you.
Okay, one last question, actually.
We have two more questions in the chat, I think.
Yeah, two more. Okay, so Devin wants to ask, wait, my fingers are, okay. How you might advise becoming involved in clinical research during COVID as a graduate student?
So once my advice, so the problem I have, and this is like this ADHD component, is the lack of focus sometimes, that I want to do too many things at the same time. And this is what I tell Jumbom. So my advice is, of course I tell Jumbom, right? It's like, you know, you should never put Jumbom in front of a buffet, because he's going to overeat. He wants to try everything.
Figuratively.
But the thing is like this, to really, if you're a graduate student and you do your research project, focus on this. Like, make this a good experience and focus on it. But if you're interested in clinical research, you have to start contacting physicians. You have to explore how the clinical trial landscape is structured here locally. I mean, you can always contact Sasha, she knows. And then you can pick people and talk to them. It's a little bit try and error to a certain degree, but I have, for example, I had requests from medical students in their first or second year to be involved in clinical research. It's, you know, the research itself is often difficult because for us, we have to most likely mentor you more than you would get out of this, because you have to understand what's the disease, what do you have to look for when you go through patient charts, because you just don't know, not being a physician. I mean, I don't want to discourage you. It's the opposite actually, but it might not be it sounds very romantic to be involved in clinical research, but at the end it can be very painful and not very fulfilling. So that's why you really have to figure out what you want and what is in it for you. So just ask around. I mean, honestly, people are very open here. Nobody will say like, oh, don't ask me. Please don't.
I think Sasha just sent her email in case you want to shoot her an email and ask too.
You're very welcome to. I answer Florian's questions all the time.
Okay, the last question. So linking the point that you have experience with moving from Germany to Canada, do you have any advice for Canadian students looking to study in Europe?
Yeah, totally. You have to do this. I mean, what you did in Germany is I really think it's important to move outside of your comfort zone, to go to a country like Italy, Spain, where they have fantastic research, but maybe there's a language barrier because, I mean, honestly, barely anybody speaks English. But this is something I would do. I think this is really interesting. Not only going to the UK, which is not Europe anyway, but where you don't have a language barrier. I think it will really make you grow. And usually it's not so difficult to find a place in Europe. It's usually rents are a little bit cheaper than in Vancouver. That's the other thing. So, and the payment is as shitty as in Vancouver. So you'll actually, it will be better for you. So, but when you do this, you should maybe do this during your PhD. So we have done this with our PhD students in Germany. So each one of them we sent, because I had such a good experience in Arefeh as well, like being abroad, being in a different country. Each one of them went to either Keith Lab, or we sent, Sarah was in the US in Stanford. We have a collaborator in Sweden. So two of them went to Sweden to work in Lund, in Gothenburg. So this is something, I think this was really good. But maybe you can do this as part of your PhD and get a scholarship for that.
Okay. Sounds great. I will look into it. Even me. Okay, so I think we have Nasim. She's asking, she says, hey, Florian, is there any way that we can make HRP and your lab closer to each other? Yeah, so this is something we have been discussing actually in the past quite a bit, how we so the bottom line is so when you do a clinical trial, you can get data or samples which you then can use in the lab. So this is the flow back to the lab. But what I would be more interested is to set up, based on the drugs we work in the lab, to bring them over to phase 1 trials here, so which are then managed by HRP. I mean, this is what I'm really interested in, and we have fantastic work being done by also Cheyenne, and she's been has done really fantastic work, and all these things she's doing, and Ali's trying to do the same thing, is all these things are published, but they're not getting transferred to like patients here, or like in terms of a clinical trial. And this is something where we really have to work and find the right infrastructure, because we need money, we need time, we need people, we need all these things for that. So that's something I would like to raise money from the community for, to make this happen. Okay, awesome. So I guess this concludes our session for today. Any closing remarks, JB? And also Florian and Arefeh, thank you for joining us. I think Arefeh needs a spotlight by herself in the future.
Yeah, I'd just like to thank everyone for coming, especially Florian for giving us a great interview. And Sasha as well, for being such a great guest.
Nobody knows who she is, right? She's like just Zoom is like these Zoom bombers, right? That's a little bit.
I zoom in, I zoom out like a ghost. I'm here just as a cheerleader.
Yeah, it's just fantastic job.
So a great addition to this interview, actually.
And we will send updates on both surveys and a video link that points to the same interview if you want to watch it again. And so thank you everyone for coming. And
thanks for listening, and thank you for your questions, right? I appreciate this.
We learned
Thanks
a
a
lot.
lot.
Oh, thank you, Florian. Okay.
Bye-bye, guys.
Bye, everybody. Have a nice afternoon. Thank you.